In this video, we will discuss about this in vitro GlyTR2 targeting TN antigen result. So here the GlyTR2 is designed to target tumor-associated carbohydrate antigen, one of the tumor-associated carbohydrate antigen, that is TN antigen, which is overexpressed on the solid tumors, for example in pancreatic cancers. However, the cell lines that they have used for in vitro testing is triple negative breast cancer. They modified this triple negative breast cancer so that it can express this TN antigen. So we will go through the results in the sequence wise. So first, in the panel A, they have shown the design for this GlyTR2. Here they have used the CD301, which can target this TN antigens, and on the other monoclonal antibodies is CD3, which can target the T cells. So this is based on this BITE design. You can refer to the BITE design videos to understand or how these BITE design antibodies are made, and they are linked together through this linker. General linker that is used is glycine serine linker. However, I have not checked the sequence in this for this BITE antigens, what kind of linker they have used. So now let's move on to the panel B, where they check whether it is expressed in the monomer or multimers or dimers. So they are able to find out that it is predominantly expressed in the form of monomer, and they then again validated this GlyTR2 using the silver stain and western blot. Thereafter they check its binding specificities in these CD3 jurkat cells. Jurkat cells are naturally COSMC gene negative. So how this COSMC gene related to this GlyTR2? So when this COSMC gene is suppressed, then it will cause high TN antigens. So the CD3 negative cells indicates that the CD3 parts will not bind to the BITE. So binding is more specific to the CD303 bindings. So the CD301, not CD303, CD301. So the CD301 bindings, if GlyTR2 binds to the jurkat cells, it is mainly because of the CD301, because the jurkat cells also does not have CD3 on its cell surface, and in order to compete the CD301 binding to the jurkat cells, they have used soluble antigens, that is TN antigen, GalNac antigen, GlcNAc antigens. So TN antigens also has a specificity towards CD301, and also this GalNac antigen has a specificity towards 301. So they will compete for CD301 that are present in the BITE with the jurkat cells. So in that case, TN antigen and GalNac antigen, we are able to see a decrease in the bindings to the jurkat cells. Whereas for this GlcNAc does not compete to this CD301. So in that case, the TN antigen that are present to the jurkat cells will bind more. That is evident from the results. Thereafter, they check other cell lines with Gale knockout. So Gale gene knockout will prevent this TN antigen expression. So in the Gale knockout, they do not see any GlyTR2 binding compared to the wild type. So this again validate the specificities of this GlyTR2. Thereafter, they move on to the cell death assay or cytotoxicity assay. So before doing their cytotoxicity assay, they modified these triple negative breast cancer cells with this COSMC gene knockout, because once the COSMC gene is knocked out, then it will express these TN antigens. So then only it is meaningful to use this triple negative breast cancer with COSMC gene knockout for this GlyTR2. So when they used this modified TNBC with GlyTR2 and also the PBMCs from the different donors, PBMCs has these immune cells, mainly the T cells. When they combine all these three things, they are able to see those dependent cell death with increase in the GlyTR2 concentrations. And in this case also they check the specificities by adding this soluble TN antigen. So when they increase this soluble TN antigen concentration, they are able to see decrease in the GlyTR2 binding to this modified TNBC. And similarly, if the binding is reduced, the cell death will also be reduced in the same order. So this also indicates that the specificities of these GlyTR2 is towards this TN antigen that are expressed on these tumor cells. Thereafter, they did the final check where they also removed. So in this panel G and H also, what they did is they not only use the TNBC with COSM. In this case, they do not use this TNBC with COSMC gene knockout. However, they use TNBC with MSC1 deficiency, or MSC1 is expressed in lower amount because they wanted to prevent the cell death because of the CD8 that are present in the PBMCs. And in the high case, they use combination of both, that is TNBC, which has this MSC1 knockout and also MSC1 deficient, and also this COSMC gene knockout, so that CD8 does not interact with this cells. At the same time, TNBC is prominent cell lines to be used for GlyTR2 specificities, and they are able to see that when they used these modified TNBCs along with this GlyTR2 at different concentrations with the PBMC donors, they are able to see increase in the CD8 plus activated cells, indicating that in this case also they are able to see the active immune cells can be activated with this GlyTR2 based cytotoxicity, and even if there is no CD8 dependent cytotoxicity present, as we can see that in the control where there is no where there is only PBMC, they are not able to see any activated CD8 plus T cells. So this indicates that the GlyTR2 is also specific towards the TACA, that is tumor associated carbohydrate antigen against which it is designed, that is TN antigen, and it can also based because of its cytotoxicity, it can also activate the immune system of the patient or immune system of the host. Though these results are from the in vitro system, we will see what are the next steps they did to further make this GlyTR1 and GlyTR2 a prominent candidate for cancer therapeutics. So that's all for this video. If you need to understand more about these experiments in detail, you can go to the reference that has been provided in the description. Thank you for your time.